Hey everybody, Mr. Murphy here. We're going to go through the final step of photosynthesis, which is chapter 6.6 on the Calvin cycle. The good way to think about this is CO2 fixation. This is the capturing of CO2 from the air and using it as an ingredient to store the sunlight energy in a chemical form, which is glucose. This is kind of like everything we've been talking about in this chapter, actually done through a series of different chemical reactions that are catalyzed by different enzymes. So here it is. The overview here, this is showing all 6 CO2s coming in at a single sort of rotation of this. In reality, these would each be individually scooped up by one 5-carbon molecule, this ribulose biphosphate, which would then become a 6-carbon molecule, which breaks into multiple 3-carbon molecules. Stuff happens. You end up producing the molecules that become glucose. Now, in terms of the stuff happening, we're taking energy from our energy carriers, ATP and NADPH, and using that energy to perform chemical reactions, change the shape of this 3-carbon molecule to achieve basically the right structure to be able to facilitate the construction of glucose at the end. So this goes into all the details of sort of the intermediate structures and reactions and things. Some key details that we should know. Ribulose bisphosphate is the 5-carbon molecule that's scooping up the CO2, and then there's an enzyme that is facilitating the combination of this that is catalyzing that reaction. So that's called rubisco. And it's called rubisco, basically a conglomeration of, the smashing together of all these words to come up with rubisco. Anyway, so it's a very important enzyme for plants. Obviously, it's an important enzyme really for life in general because it's this enzyme that facilitates the capturing of CO2 from the air and storing that back in a form that turns it back into something that's actually useful for us. Way back earlier in this chapter, we showed how CO2 has very little free energy. So we're taking the individual CO2 molecules, breaking them apart, and using that to produce the product, which is glucose. Again, the chapter has a lot of detail about the intermediate stuff. That's definitely not relevant to us for the scope of what our course is about. But of course, may be worth understanding if, you're trying to test your sort of understanding of the broader chemistry of these things. You could follow along in the diagrams. You'll see that there's nothing fishy going on. It really does, keep track of all the oxygens, all the hydrogens, all the carbons. And this glyceraldehyde 3-phosphate G3P is the 3-carbon molecule that exits the Calvin cycle. So for every 3 carbon dioxides that are captured, you get 1 of these. Therefore, for every 6 carbon dioxides captured, you get 2 of these, which is enough to then make a glucose. This goes into a little more detail on the regeneration portion, which is turning sort of these molecules back into the 5-carbon ribulose bisphosphate. But like I said, that's a good level of detail for us. Returning to some terms that we should know, these are photosynthetic organisms that utilize this. They're also known as autotrophs. They generate their own food. I think often we sometimes forget, right? It's easy to think of plants like, oh, they do photosynthesis to get energy. Well, they do photosynthesis to take energy from the sun to make their food. From there, they're just like animals in terms of needing to break down that food through cellular respiration in order to actually use that energy. So that's an important distinction to understand. Heterotrophs, autotrophs and heterotrophs, autotrophs literally means food like from the self, so self-food. And heterotroph means food from other, so finding food from the things that it consumes, which are plants or other animals. Ultimately, it comes down to the same food that was generated by those plants. All food chains lead to ultimately plants that were using energy from the sun. Cool. At the beginning of this chapter, it presented a question about how yeast can perform in the absence of... No, hold on. It was a question that was basically saying that yeast cannot perform fermentation in the presence of oxygen, something along those lines. Anyway, when we talked about fermentation, we showed how the process of fermentation occurs when there's a lack of O2, because the buildup of NADPH becomes a problem. Glycolysis makes NADPH, it makes pyruvate. The buildup of both of those kind of hinders the continuation of glycolysis. Ordinarily, those would be shuttled away to the mitochondria and performing the rest of cellular respiration. In the case of fermentation, though, we're basically denying that as an option. We're taking away that pathway. And it turns out the way to do that is to deny oxygen. With no oxygen, the electron transport chain stops. The citric acid cycle, or the Krebs cycle, also stops because it can't keep producing the products that need to be shuttled to the electron transport chain, because it can't get any more NADP, it can't get any more FAD, NAD or FAD. So it shuts down too, and so fermentation provides a way for glycolysis to continue, and that's how the idea of fermentation works, which is the production of different alcohols. In the case of yeast and bacteria and things, can produce alcohol through alcohol fermentation. So, production of bread and things like that, we rely upon yeast, which is an organism, it's a fungi, it's an organism that contains mitochondria, but when you deny its mitochondria the ability to do the latter half of cellular respiration, it forces it to compensate through this process of fermentation, which, if you're a tiny little microorganism, you can get by on that for a while because you don't have very high energy needs. For us, we can't rely solely on fermentation. Getting 2 ATP per glucose is not the same as getting 38 ATP, and therefore it's not enough to sustain a very large organism. But on an individual cellular basis, fermentation can keep the cell alive, but of course you're exhausting yourself quite literally in the case of whether you're running or whatever you're doing, that's pushing yourself to that limit of needing to rely upon fermentation. So just a little bit more on that topic, as every chapter likes to have an opening and closing kind of question segment. So there you go. If you have questions on any of this chapter, anything at all, please let me know. And don't forget that we have separate photosynthesis and cellular respiration sort of all-in-one videos as well. Thanks.